It's with great honor and pleasure that I introduce our keynote speaker, Stephane Bancel from Moderna. He's going to be sharing the science of mRNA. This is the man that from our first panel, we were talking about selling that dream, and he sold that dream in the largest biotech IPO in history. And he's right here in Cambridge with us today. That dream is becoming reality, hopefully. And a leader who has experience in large pharma, is an advisor at several groups, is a venture partner at Flagship, keeps everything alive with serving on the board at the Museum of Science and pumping innovation and thinking creatively and openly. Really, really distinct pleasure to hear from this dynamic leader in our town. And thank you, Stephane Bancel, for being here tonight.
Well, I'm all red and blushing now. Thank you for the kind words, and good evening, everybody, and bonsoir. I'm delighted to be here, and I'm very honored to be here. Thank you for having me tonight. What I plan to do is to tell you a little bit about why we got excited 8 years ago about Moderna, and why we built the company the way we built it. As you'll get a sense, it's a bit different from where most biotech companies are built. And we did that because of the science and what we think it can do for patients. So let me start by doing the usual disclaimer. I'm going to be quick, but I have to now. So I'll be making forward-looking statements. Making medicines is really risky. Making medicines with new technology is even more risky. We don't have commercial products, and you can look at all the risk factors on the SEC website or on the Moderna website. So now this is done. So I had the chance almost 8 years ago now at the flagship office one evening. I got a phone call from Noubar Afeyan that some of you know. I was at the time running bioMérieux, and he called me and he's like, where are you? Because I was always traveling around the world because bioMérieux had subsidiaries in 40 countries. I'm like, actually, I'm in Boston today. And he's like, well, you need to come to see me before you go home. And so at the end of the day, you know, the cold winter Boston day, I walk to Flagship, and he shows me data of one experiment that had been done at Harvard, showing that he had used messenger RNA injected into the muscle of the mice, and they found human EPO because the mRNA was coding for human EPO in the mouse blood, and it seems to be active because the red cells were through the roof. And so given what I had done before when I was at Lilly or in grad school, I told him, this is really, really cool. It most probably will never work. So thank you very much. I'm going to go home, have dinner with my kids. I told him using messenger RNA seems like a really, really silly idea, to be polite, because of course we all believe the mRNA was highly immunogenic, fever, vomiting, not so cool for a drug, and we all thought mRNA was very unstable. Not so cool for a drug either. And so Noubar was very good. Instead of trying to convince me, he told me, well, you're going to have to make your own opinion. I believe this experiment is correct. My team went to Harvard and they checked the lab. It is being repeated as we speak. But what if this is true? I'm just asking you to think for 1 minute, what if what I'm showing you is true? That one day we can, by working through technical challenges and so on, inject mRNA in a human, and that using the patient ribosome machinery, you can make literally any protein you want. What if this is true? And I told him, if this is true, you're going to be able to make a lot of drugs that you cannot make using recombinant. And for the non-biologists in the room, it's because, does that work? Yeah. the biotech industry for 40 years has done a wonderful thing for patients using secreted protein, making secreted protein. But 2/3 of the human genome is coding protein that are transmembrane, that are attached to the membrane of the cells, or that are used intracellularly for the functioning of the cell. Around 2/3. So 2/3 of 22,000 protein ballpark. That is a big number of proteins that we have not been able as a society to drug for patients. So I told him, well, if this is true, you're going to be able to make a lot of product that you cannot do using recombinant. And then we talked a bit about the science, and I say most probably you're going to be able to combine different mRNA in a single vial. Think about it, in our cells, we have millions of mRNA at the same time in water. So if nature can do it, there might be a way. It might take us a long time to figure it out, but there might be a way to do combinations. And that was the first point. So a very, very large opportunity to help patients. The second one was probability of technical success. Having worked at Lilly for 4 years, I had the chance in the front row to observe what we all know, which is most drugs that are going to the clinic won't make it to market. And the piece that was very interesting about the idea of using messenger RNA as a drug is that if it was going to work in one application, pick rare genetic disease in the liver, pick vaccine, pick any application you want, the second drug that was going to follow that first application, if the first one worked, the chance of a second drug working was very, very high. If you think of a small molecule, there is no correlation between the one drug and the next drug in terms of efficacy and safety. But here, if you think about it, because messenger RNA is actually an information molecule, it's made of 4 nucleotides. If the first application works, let's say to go to the liver for genetic disease, the second drug is exactly like the first drug, but the order of a nucleotide on a message. The chemistry is the same, the manufacturing process is the same, the formulation is the same. So the first one works, the second one coding for another human protein, which, by the way, we are not inventing the proteins. Evolution has done that for us. This is the second part of a probability of technical success, which is on the one hand, you have a platform, because mRNA is an information coding molecule. It's never the drug. When we inject mRNA in human, we never inject the drug. We inject an instruction set for the patient's ribosome machinery to read it and to make the protein we just designed. So we thought, in that first evening, we discussed, geez, if this really could be true, you could have very high probability of success versus traditional medicine, because you will have a platform, so you'll have read-across product, and the product will be made by humans, is a human protein, like if they were healthy. So this is pretty cool. The third thing we talked about, and I told Nubar, well, if you were to invest in process development to really understand the manufacturing process really well, because it's always the same, it's a platform. If you have to invest in robotics and IT a lot, you should be able to grow really, really fast. Because unlike what I observed at Lilly, where sometimes we'll wait quarters for the engineers to figure out how to make the product, because some of those products are very hard to make. So you can start a clinical trial. So we should be able to fly, if we have invested a lot of capital, to be able to just industrialize making always the same thing. And then, because I'd been away from the science of mRNA for a long time since grad school, I asked Nubar, by the way, how do you make mRNA again? I forgot. How do you make it? And I'm a biochemical engineer by training, so he knew how to speak to me. And he's like, it's liquid phase, cell-free manufacturing process. So I said, oh shit, sorry for my French. It's going to be really, really cheap. It's like, yep, no cells to make it, in water, enzymatic. The more you concentrate the solution, the more you're going to make product. I'm like, wow. So I told him, so you tell me that if the thing we're looking at, that one slide of data from Harvard is true, we're going to be able to make maybe dozens or hundreds of molecules over 20, 30 a time frame, not 6 months. That will have a higher chance of working. We can go faster than traditional pharmaceuticals, and that will be cheaper. He's like, yes. I'm like, what am I missing? Because that just seems so good to be true. What am I missing? And he's like, I don't know, I've been thinking about it for a month with my team and we cannot find a hole. If this data is true and replicated in primate and then in humans, this is a new class of medicine. So after thinking about it a little bit and with a bit of homework, because it was a crazy idea with one employee, and $2 million of cash in the company, I decided to leave bioMérieux and resign from bioMérieux. And in the summer of 2011, I left bioMérieux, and a few days after, I was at Flagship in an office with a white page of paper saying, okay, how do we build this company? And so the piece that has been very important to how we build the company, which sometimes some of our friends like to use funny names to talk about us, is we started with that belief that if we were right, if we could make the science work, this would be a new class of medicine, having a massive impact of patients over the next, again, 20, 30, 40 years. So we said, it makes no sense that this will work for only 1 or 2 drugs. It's either 0, because we are wrong, and we cannot figure out before we run out of money how to make it work, or it's going to be dozens, if not hundreds, because anything else makes no sense. 2 or 3 drugs make 0 scientific sense. It was 0 possible, or hundreds. And so we said, we're not going to build a company assuming 0, because then you stay home and you do something else. So we had basically the strategic imperative, and it was actually very easy at the end of the day if you think about it, is we had to go big. Either you do nothing and I stay at bioMérieux, or you go big. But going for 1 drug just made no scientific sense, and it was actually very risky. And so we said, if this could be a new class of medicine having such a big impact on society and patients over a long term, we need to be totally paranoid and to overthink risk. And that's what we do all day long. If you were to come and be a little, fly on Moderna's wall tomorrow, you will see the teams walking around and interacting and talking together, and we'll be talking about risk all the time. And the 4 risks we talk about all the time is obviously technology risk. If you do a new technology, don't be surprised, you have massive technology risk. So what we've done over time is trying to reduce it by investing in science, by trying a lot of things in parallel. We always say there's always the unknown. We can do so much in animal testing. We might take in a clinic if we only take 1 drug in 1 application. We might take the 1 thing that we didn't know is not going to work. If it doesn't work, everybody is going to believe mRNA will never be a drug. Employees, directors, investors, the media, everybody. Not going to be very helpful. So we said we have to manage technology risk by trying a lot of application in parallel. And then we said for every application we're going to try, if we only pick 1 drug, we might have a biology wrong. If you have a biology wrong and the drug fails, it's still failure. Everybody's going to assume it was not the biology but the technology, because it's such an easy way to think about it. We as humans like to have reductionist thinking. I'm the same, don't worry, I'm very, very biased like everybody else. The third one was, of course, execution risk. Every enterprise in the world, any size, has execution risk. And the 4th one, because unlike bioMérieux, where every night I had a chance to go to bed with more cash than in the morning, here at Moderna, for 8 years, I have a chance to go to bed with less cash than I had in the morning. And now it's around $3 to $4 million a day. So every night I go home, like tonight, we're going to be $4 million into a balance sheet tonight. So we obsess about those 4 risks. Of course, there are many more risks. And if you want to have a sense, again, read the S1 on the SEC website. It has a section like this of risk factors. But those are the 4 risks that I believe are the most important. And I believe our job as a team is to manage those 4 risks and do everything we can to reduce them as much as we can. So one easy example, which I won't talk about tonight, I'm happy to take questions, is financing risk. One of the ways we manage financing risk over the years, it's simple to say, you guys are going to smile, it's really hard to do, is have a strong balance sheet. Always try to get 2 to 3 years of cash, and as your cash burn grows, just keep raising even more money. Because if the market tanks, like in 2008, which I had the chance to run bioMérieux at the time, and everybody goes down, private market freezes, public markets look ugly, you don't want to break the company because you didn't have enough cash. So we said we like having a couple years of cash. Again, it's super easy to say. It's not so easy to do. So let me talk to you about 2 risks that I think explains why we build Moderna the way we build, and I hope as you go back and if you were to look at the company or press release or whatever, what I'm explaining will make some sense. So we say technology risk, which we put on the x-axis, and biology risk. So again, this is all our assessment. There is no book I can go buy at the MIT bookstore to figure out mRNA technology risk. That book does not exist. So it's the team and I, or advisors, trying to guess and talk together and spend a lot of time working together to figure out what do we believe is less risky, what do we believe is more risky around the technology. And I will use a couple examples. And then same on the biology. I cannot go to Harvard or MIT buy a book about biology risk. It goes back to our own assessment. So we started the company with vaccine in the clinic, which a lot of people commented that this is everything else might be not working. That's why they're going to vaccine. Actually, we went after vaccine because we thought, after playing with the technology in the labs for a year or two, that it was most probably what we knew at the time, the least risky thing to start a human trial on. And the rationale was the following. With vaccine, you need 1, 2, maybe 3 doses, depending on the vaccination schedule, not dosing a kid forever like in a rare disease. So that seems less risky, a couple doses than a weekly dose forever. With vaccine, you need very small doses of material because the immune system, once you prime the immune system, you have amplification through the B cells. So you get a nice leverage from nature, which is always good in biology when therapeutic index is what kills drugs, or lack of. And the third piece was we did not have to code a human protein in the coding region of the mRNA. We had to code a virus protein. And we thought that was very important for safety. Because once you go in the clinic first, if you don't know what you don't know, and you take a group of healthy subjects and you give them an mRNA, let's say for human EPO, which we made plenty of human EPO mRNA over the years, and you have something you did not anticipate in animal studies, and they mount an immune response to human EPO, you're going to get those healthy subjects sick forever. So we said this is not a good idea because we don't know what we don't know. And so as we spend a lot of time learning all those things and thinking about all those things and really agonizing about which one do, we say vaccine is most probably the lowest technology risk we have with what we know now. So we started with vaccines. Because we thought the game was to go one step at a time, not try to run for a marathon, to start in the brain. We thought it was less risky to do that for patients and to de-risk the technology in the company. And then on biology, we had the same thinking, which is, do you start with HIV or flu? And we say, we're going to start with flu. It's not going to be very sexy, but flu has an FDA-approvable endpoint because of seasonal flu. If you can show the FDA or scientists and clinicians or investors that you can get a 40-to-1 antibody ratio, that will get you an approval for seasonal flu vaccine if your platform is approved by the FDA. So we say that's cool because it means in the phase 1, we can go dose healthy people just in the intramuscular injection, and we'll know very quickly, i.e. months, not years, like some phase 2 will take you. Are we home? Are we there at the 40-to-1 ratio? Are we a log away or 2 log away? We had no idea. We had never run a human experiment. So that's why we started with flu. And so we started with flu, and then because flu looked pretty good, and we're totally paranoid, as I told you, so we did it twice with 2 different flu strains. We did H10 in Germany and H7 in the US. Just to be sure, and both looked amazing. They have been published now. H10 showed us 100% of a healthy subject had more than 40-to-1 the FDA approval endpoint concentration in their blood. The H7 in the US was 96%. One subject was at 20, not at 40. So that's pretty good for first time trying. So then we went after a big commercial opportunity that we believe are going to be important for patients, which is why we get up in the morning. With CMV, cytomegalovirus, this is the #1 issue of birth defect in the developed world. There are around 10,000 kids in the US alone, larger number in Europe, that are going to be born with mental disability or be dead before birth or dead a few days, weeks after birth because their mother got unlucky. She got infected when she was pregnant with CMV. There is no vaccine on the market. I'll go to the biology in a few slides, why there is no vaccine on the market, because the biology is really complicated. And we use that also once we got the interesting data that we could elicit T-cell response with a vaccine in human. That maybe it would be worth trying this in cancer. And we went after doing a personalized cancer vaccine. So if you think about risk, this is borderline off the chart. Last time Tal did a count, he counted more than 1,000 clinical trials for cancer vaccine over the last 30 years, including industry clinical trial and academic clinical trial. They've all failed. So the odds are not really in our favors. So this is why we partnered with Merck and Keytruda to do it. But this is a personalized cancer vaccine. So I could spend the whole evening talking about this, and I'm happy to take questions. The concept is very simple, but it's a beast to do from a manufacturing standpoint. We take a biopsy of a patient, we next-gen sequence it, we next-gen sequence a healthy cell of the same patient, we send it into the cloud into an algorithm we developed with key academic and clinicians around the world. We used to select 20 antigen, now we are 34 in the current version in the clinic. We drop it down, we make a product from one human being at a time. With 20 historically, now 34 antigen that are specific to your tumor. We send it back to the clinical trial site. From needle to needle, from biopsy to injecting it, it's around 50, 60 days. 50, 60 days to make a product for a human being. We had the idea of doing this 3 years ago, and now we have been in the clinic for 14 months. And we've shown in the clinic that we can elicit T-cell to the antigen of those patients that have been dosed with the product so far. So then we try something else. We say, there's a very cool feature of mRNA that the biotech industry has made us forget, which is a lot of protein encoded in the human DNA are not secreted protein, they are paracrine factors. They are used in your body to make protein locally. So one of them is called VEGF. VEGF's job is to grow new blood vessel. When you cut yourself, locally your body is going to express VEGF to draw new vascularization. So there's a professor at Karolinska who had been working all his life on VEGF, saw the technology, and made a very cool drug, which is now in phase 2 trial in Finland, where they inject our mRNA drug. It's in water, so it's mRNA in water, there's no formulation, in the heart of people after a heart attack. We have shows in pigs, and the phase 1 data was very encouraging. It was published in Nature Communication a few weeks ago. They showed in pigs that they can improve survival of pigs, that they can improve ejection fraction, the ability of the heart to pump. Because if within 48 hours of an MI you inject our drugs in a pig, we don't have a human data yet of that experiment, it's ongoing. You increase the survival of the pigs, and as you sack the pigs and you look at dissections of a pig's heart, you can show you reduce fibrosis and you grow new blood vessel and new tissue. So that's where we started to inject directly a drug into an organ. And the other idea then we tried with a formulation is to do the same for cancer. So we do a personalized cancer vaccine, which is one way to try to go after cancer. We have a totally orthogonal approach to cancer, which is, what if you injected some mRNA into a tumor? Could you improve the response of a checkpoint alone? As we know, the checkpoints, PD-1, PD-L1s, have been wonderful for many, many patients. When they work, it's like a miracle. But the problem for patients is there's 60-70% of patients, depending on tumor types, for which they have no response. They still are tumor types like ovarian cancer where PD-1s are not approved because they have failed in the clinic. And so the question is, could you use mRNA, and what's unique about mRNA, to inject it into a tumor, to use the tumor cells as a little factory, to do something very specific in the tumor that doesn't have a systemic effect, so you don't have a tox profile. And so we went with this first product, OX40, which is now in phase 2 for ovarian cancer because we got some interesting phase 1 data where you saw lesion regression. Is injecting OX40 is a human protein whose job is to recruit the T cell. That's his job in life, is just to recruit the T cell. So we inject mRNA coding for OX40 into tumors. The tumors make around 1,000 copies around 48 hours time frame. And those proteins are membrane bound, meaning they are on the outside of the cancer cell. They're attached to the cancer cell because they're membrane bound. So the way I describe it to my kids when they saw the photos of the patients, they said, geez, how does it work? So I had to explain to my little girls. And I said, the way I think about it is like decorating the cell like a Christmas tree. You have around 1,000 copies of those proteins on the membrane of the cells who basically tell the T cells, hey, come here and eat me. I'm right here. That's what OX40 ligand as a protein do. And then we went to a new product, which is much better, because the cool thing about this technology is the more scientists think about it, the more they come up with crazy cool ideas. They say, well, if we could do a PD-1 commercial systemic, add OX40 to recruit T cell to the tumor, the way to really kick up the response is mess up the immune system by putting a bit of gasoline on the fire, by putting IL-23 and IL-36 gamma. And the data have been published a month ago. The data are really super cool. So the idea is kind of crazy drug. It's actually 4 drugs. It's an IV of a commercial checkpoint like Keytruda or PD-L1, pick your favorite checkpoint, and you inject in the tumor OX40 ligand to recruit T cell, and you create a bit of gas on the fire with the 2 cytokines. So that's in phase 1 in the US. We cleared the first dose, we got the OK, and we started dosing a patient at the second dose. So that should be interesting data coming in the coming months. The last place we are investigating in the clinic as we speak, which I think will be in the next 12 to 24 months, a very exciting data we're going to read out, is our ability to use the liver as a factory by going IV. So this was intramuscular, the first 2, intramuscular, directly injected into the heart or here a tumor. And here, those 2, because there's another one coming, is by going IV. So the first one here, the 5th vertical of the company, is using the liver. You don't have to take photos. You can have a very nice slide on our website. They're all on the website. If you look at the protection, we did our quarterly earnings call last week. They're all on there. You can still take photos, but they'll be better on the website. The idea is, can you inject IV and mRNA where most of the cargo will go to the liver and use the liver as a factory to make secreted protein? This is basically anything that the biotech industry do, you can do using this technology, just this vertical. You could do EPO, Herceptin, pick your favorite recombinant, PD1, anything like this. So we started with an antibody. Why? Same strategy as always. Start easy before you go complicated. Start easy before you go complicated. So this is a program co-funded by DARPA, the Department of Defense. They want this as a proof of concept of could we do very quickly an antibody response as a treatment in case of a pandemic. Because, as I told you, with personalized cancer vaccine, we can go in around 50, 60 days from a sequence to a product QC approved to ship somewhere. So it doesn't seem crazy that one day there's a new virus, protein C virus coming. If you give us an antibody, then that's the vision that the Army has. They gave us that grant. If you send us by email or through a secure website the sequence of an antibody that you isolated from a human, we can give you really quickly antibodies that you could use as treatment. Very, very quickly. Quicker than even you can do your tox study with a recombinant technology. Not because we're smarter, just because of how our technology works. It's a platform. So we're starting with chikungunya antibody. This is in the clinic. The first dose has dosed. We're going to do dose escalation. And this is the same technology that is shared by those 2 verticals. This vertical, the difference, this is for intracellular. So again, we start with secreted protein, and then we're going to, this is a rare genetic disease, MMA. It's a metabolic disease. The kids who have that disease, because they got unlucky with a DNA lottery, they have massive brain and kidney damage because there's a protein they are missing. It's called the MUT enzyme in their liver. For the scientists, in the mitochondria of their liver. And we published in Cell that when we make the mRNA, I'll show you the data in a minute, you inject IV, most of it goes to the liver makes the MUT enzyme, and the way we code the mRNA, we tell the liver cells, that protein you just made, now you take it inside the mitochondria of your cell. And it does. The data we published in Cells with the NIH, because they ran the model, we did not have a model, shows that 100% of the animals on placebo were dead after 5 weeks. 100% of the animals on our drugs were alive 12 weeks out, which for a mouse is a long time. So this is going to be tried in the clinic very soon. The IND is now open. It was opened by the FDA in 30 days, actually in less than 30 days. We have a fast track designation from FDA. The team is working really hard to get the sites up and running. We did a national history study in the last 18 months, so we start to know where the kids are and to monitor their biomarker to help us later for approval. So we have a baseline because, of course, we will not run a placebo-controlled study. That would be unethical. So we're going to use the data of each kid before they get onto the trial to get a baseline and also how disease is progressing. So that's how we've been thinking over the last 8 years about managing technology risk and biology risk. We did not decide to go after one drug. We thought going after one drug was crazy because we could fail society, because if we picked the wrong technology application and or the wrong biology, it will be a negative clinical readout, and the entire planet will think mRNA cannot be a drug. So that's how we started it. This is, for the scientists in the room, one of my favorite Moderna slides. This is what I call the Moderna pipeline for geek. So this is the current 20 drugs we have in development. We do not talk about what is in research. This is just what is in either the clinic or in talks. I'm just going to highlight a few to give you a sense. This is the vaccines. So we have now 9 vaccines. This one is super cool. The CMV is a 6 mRNA product. 6 mRNA product for the vaccine. 6 mRNA in a single vial. This is 5 proteins together coming as a structure. This is a little ball of protein. It's a virus-like particle. This we made as well. Here you see the MMA I talked about inside the mitochondria. They just give you a sense of the things we can do. The only reason we don't have 40 drugs on that slide is I don't have enough money. That is true. That is the only reason we don't have 40 drugs on that slide. There's around 10 drugs right now that are in waiting mode, like the airplanes above the airports, ready to go into talks. We don't have enough money to pay for it. So this is the Moderna pipeline for investors. So I have to do that slide as well. What is in red is what has changed since the IPO, which was 90 days ago. So in 90 days, we had 2 drugs moving from positive phase 1 data to phase 2 planning with file to the FDA for phase 2. A new positive phase 1 data for a combination vaccine that doesn't exist on the market for 2 pretty nasty respiratory viruses. 2 new drugs starting in the clinic, 2 more open INDs in 90 days. So I won't go through all the vertical data. I'm going to skip a few slides. I've been given the eye and the sign. But as you look at the company, as you look at the data and the clinical data we published, the way we think about it, we think there are 3 big questions. Is the mRNA well tolerated? Of course, first question. The second is, does the mRNA we make encode for the right protein? And then is the protein giving you the pharmacological effect you are looking for? What we have shown in now 4 of those vertical, those first 4 in humans, yes, yes, and yes, and multi-times. These 2 is what we're going to discover together this year as we run those clinical studies. So I'm going to skip all the slides on the internet. Don't worry, you can go read them. This is a summary, vertical by vertical, of the key program and the data. I just want to draw to a close before taking question. We have 3 priorities now. The first one is to move a pipeline forward as fast as we can, because a lot of those medicines are super, super important. Most of the medicine we do, there is no alternative for patients. In a rare disease, in a vaccine, I don't have to explain that cancer is important to anybody in the room. The second priority is to keep bringing new development candidates. Now that we spend years building the platform, investing $100 million per year just on science, not on drug discovery, on science. We spend another $100 million per year on drug discovery. And we have a big factory now in Norwood, Mass, 10 miles south of here, that has been built and is up and running. So getting new candidates moving forward. And the third one is we want to keep inventing new verticals. We believe with those first 6, we're just getting started. We have a team at Moderna whose only job in life is to keep inventing vertical. Their track record has been pretty good. We have done 6 so far, and the data so far has been pretty encouraging. This is a summary of a company in a few numbers. Maybe quickly on the second row. So now we have 5 immuno-oncology drugs in development. 3 are in the clinic. 2 have open INDs, and we'll dose very soon. 1 is partnered with AZ, 1 is partnered with Merck. We own the other ones. 4 rare disease program with first IND open. 3 vaccines in the clinic with, for 3 very large unmet medical need, where there are literally thousands of people every year suffering because those vaccines don't exist. The team is now 750 employees, so we've grown a lot. We're still growing this year. I think there's 150 jobs open for the year. Norwood was a $100+ million investment, 200,000 square feet factory, and the balance sheet is still strong, $1.7 billion of cash at the end of the year. So what if mRNA could be a new class of drug? And the piece that's most important to us is why we do that. If you think about the type of things I've already shown you, if you think about the things we think we can do in the labs and soon in the clinic, I believe we can barely imagine what we're going to be able to do the next 20 to 30 years. More than half the things I showed you on the pipeline for geeks, that's a nice slide with all, but when we started, we have no idea we could one day do that. But when you put this very amazing technology, because mRNA is information, when you put great scientists around it and you give them the capital and you give them the freedom to do it, they do amazing work. So thank you for your attention, and I'll be delighted to take questions. You want one microphone?
Thank you very much. Very impressive presentation, and obviously very exciting for all of us scientists here. Questions regarding the nanoparticle you're using for delivery. What kind of long-term tox data you have, in particular in the liver? And are you exploring the idea of exosome, using exosome or stem cell exosome as a more reliable way to deliver? And if you are using exosome, how do you make sure they are coherent in terms of molecular entities, it's the same way, and so on?
Yeah, so we don't comment about the topics we are doing in research, so I won't talk too much about exosome now, but we are aware of exosomes, as you can imagine. In terms of a lipid nanoparticle, when we started with kind of the state of the art of what has been done in nucleic acid, and we thought it was not ready for the clinic in our eyes. The piece we didn't like about it was the half-life of the lipids. They were, in our opinion, way too long. And so we basically went back to the drawing board and invented new classes of lipids. We invested a lot in chemistry and formulation technology. One of the lipid for the rare disease, which is the one because of the repeat dosing that we think this is very, very important. We presented the data in a few papers, and they are in our S1. The half-life is around 2 hours. So they literally fall apart as they get into cells, which we think is really, really important. And we had to re-engineer everything around the surface chemistry of the lipids, because as you can imagine, when you inject a lipid to the immune system, it looks like a virus. The size is perfect, so anything on the surface, the immune system doesn't like it so much. So basically what we realized is that if you use old technologies, you have clearance problems that was not appropriate to do medicine like we wanted to do it. For one of the applications, for the 2 verticals on the right for IV delivery, we had at some stage around 18 months setback, where basically what we were working on, when we decided to do repeat tox in primate for a long time, it didn't look so good. And so we said, okay, we need to go back to the drawing board. The scientific team did an amazing job trying to really understand what was going on. And that's, again, the thing that I think we have been very fortunate is because of the capital we have. We have around 300 scientists today whose only job in life is to work on the technology. They don't do drug discovery. They, of course, collaborate with the teams doing drug discovery, but those 300 scientists, they work on inventing new lipids, all the formulation components, even process. We realized as we learned about this technology that none of us knew when we started, that manufacturing process is extremely important for the biological properties of the lipid once in vivo. It's a great question. It's not easy.
Thank you very much. A very inspiring presentation. My name is Vanina Laurent-Ledru. I work for Sanofi. A question on something that I'm sure you are starting to think about, value and access. If you can explain a little bit what is your current thinking. And also I'd be interested to understand how developing countries are seeing the approach, because I imagine that it can be a very attractive approach for them. Thanks.
Yes, thank you. It's a wonderful question. So if you look at the pipeline in detail, I think you will observe that most, if not all, of the drugs that are in our pipeline have no competition. And that's why we pick those, because we believe that, given as I shared, and it was not a joke, that we don't have enough money, so we are very, very disciplined to say, look, if there's a good drug, with another technology, what's the point of making another one? When we started to work on the intratumoral oncology approach, I told the team, don't come back telling me you want to do another PD-1 because I'm going to fall asleep. I have no appetite to do another PD-1. There are great PD-1s on the market. I don't see it's a good use of our capital or on time on this planet to make another PD-1. So I think we're going to try to be very practical about what value do we think the drugs can deliver to the healthcare system. On access and on emerging markets, as you can see if you spend a bit of time on our pipeline, I don't have time to go through it. We think that we have a very important societal responsibility to make the platform available for poorer countries, which is why we have around 5 vaccines that have no commercial futures, like the chikungunya vaccine, we have a Zika vaccine, we have flu vaccines for pandemic region and so on. And the way we have thought about it is to say, we want to make the platform available for the common good. And what we need to find out is capital, that at this stage it's not a shareholder capital, because the next time I come back to ask for more money will be a tough discussion. But to partner with private foundations, like we've done with the Gates, and with a couple, it's a public information HIV project with the Gates. Partner with governments. The BARDA, which is part of HHS, has given us a 125 million grant, and they are paying the entire development of a Zika vaccine. And so we want to continue to do that. We have actually somebody from Sanofi, Mike Watson, who is actually spending a lot of time in planes, going around the world asking for money, really for those type of projects, which is, there are still too many people around the world suffering from infection that we could have an answer for. The beautiful thing about this platform and this technology is that you don't have to spend the same type of money than the traditional vaccine manufacturers have to spend just because of how the technology works. And so we want to partner with private and public organizations to find the capital so that we can address those issues. What happened several times, and it's happening as we speak in Africa around Ebola, should not happen. We have a beautiful Ebola vaccine that is still in preclinical testing, ready to go to tox. The challenge model done in the US Army Library is fantastic. I cannot find money to get it to a clinic. So that's something really, really important for us. Yes?
Thank you, Marcel. Stephane, it's a great presentation. As you may know, the first presentation was on the IPOs.
Yes.
I'd be curious to kind of get your perspective on what the experience was for you. And then one specific question about who drove the process, the management team or the investors? And the decision to do the process. Again, there was a bit of a discussion on earlier today.
So first we were preparing the IPO for a long time. We tried to stay private as long as we could. We did pretty good. It was 7 years private. If I could have stayed 5 more years private, I would have done so. The chance I had is that I had run a public company before, so it had never been a goal to go public. It was always a means to an end to build the business. None of the pre-IPO shareholders sold 1 share into the IPO. It was only primary shares. And so for years, I knew that one day we had to go public because the private markets are so deep. I think we even surprised ourselves that we could raise so much money privately. Again, if in 2011, when we started thinking about this technology, you had told me, Stephane, you're going to raise more than $2 billion in the private market. I said, yeah, great joke. I have no idea I'm going to do that. It has never been done before. So as we did every round, we kept increasing the investor base with traditional public market investors. So it's now public information that Fidelity has been an investor for a long time, since December 2014. Pictet has been an investor, Adia has been an investor. So we just, at every round, brought investors that could do private, but whose core business is to do public. So we prepared the IPO like this, and then, we decided to go public because it was our time when we did the last private round in January of 2018. We literally finished our global tour because for the first time we brought investors from Singapore and China and Middle East with Adia. And I'm like, okay, I don't know where I can go anywhere else with a lot of US investors, European, and now it's almost been around the world. So I'm like, I think we're done, or what we can do privately. And so we had always indicated to investors that we'll be patient and that we'll go out when we had enough human data to help us kind of raise enough money and build the company. And so it has been a dialogue with key investors, maybe. If you do the math and you look at the public information, the top 5 investors of a company control 30-ish% of the capital. So you can literally place a couple of phone calls or have a very quick conference call to kind of talk about the big stuff about Moderna. And the largest shareholder is still at the board, which is easy. The second largest investor is still at the board too, that's easy too. So it was really a dialogue, but we are always clear that people have to be patient. I remember when some investors like Fidelity or Viking invested in at the end of 2014, we told them we have no idea we're going to be private 4 years, 5 years, 6 years, we have no idea. What we told them is we want to build a business for the long term. We want to invest in science, and because it's science, you have no idea if it's going to take you 2 months or 2 years when you do something. And we're trying to build a new class of medicines. We're going to be patient. We're not going to try to rush to the clinic if it's not ready, which is why, for example, when the old class of lipid didn't work to our satisfaction, we took a step back. We, of course, communicated to our investors in real time and so on. So it has always been kind of a long-term process. We've always had the mindset to build a company for the long term that could be a key player, ideally the leader in a new class of drugs, and to say we're going to try to find investors who understand what we're doing. The trade with that is that to get some of those investors to invest, for some of them, it's taken us months and weeks of diligence. Because at the time we didn't publish a lot, now we publish much more. I think there's around 16-ish papers on our website if you want to go look at them. We don't have a lot of IP issued, so you have to go look at the IP in detail. And a lot of the technology was not as shared or understood, so we had to spend a lot of time with people. Adia invested last January, a year ago, and they must have spent 6 months. I must have gone to Abu Dhabi 3 times. And they have sent teams in Boston to talk to our scientists and our clinicians. So it's been a very long process, but that was we're always very clear who we were. We're not trying to please investors that didn't fit our strategy. So if somebody didn't like what we were doing, because they say you should do one drug and forget the rest, okay, thank you very much. We don't want to do that. Go invest in a one drug company. We want to build a new class of drug. You need to believe this could be dozens and dozens of medicines if you are patient. If you want to double your money in the next 2 years, go buy something else. So we're always very true to ourselves and always try to find investors who shared that vision, had the chance to witness the execution. Sometimes investors will pass in a round. We'll still talk to them and keep talking to them. And sometimes they invested the round after. And sometimes they invested the round after. We had people coming into the IPO on the night of pricing the IPO that had never bought private, but we talked to them for 5 years. So what this gave us as a huge benefit is they had seen what we told them 5 years ago, and they're seeing along the way the execution, the setbacks, everything. So they knew what they were investing in because we spent so much time building the relationship and the trust. We could not have pulled an IPO like this by just showing up at people's window, even in TTW, say, hey, we're going to price in a month or 2. It doesn't work like that. I want to be careful. I'm sitting between you guys and wine, so.
Thanks for the lecture. I'm from Harvard Medical School and I'm also working on DARPA projects on infectious disease. I want to hear about your insights on drug or therapy development for infectious disease like Ebola, HIV, or flu infection kind of stuff. Because according to my understanding, all the antibodies, drugs, or therapies are all passive solution because they're responding to those virus or bacterial infections. So, but if we look at the nature, like bats, mice, or rats, they're living with those bacteria or viruses, but they're like tolerant to those infections. So instead of we like update our drugs or vaccines or antibody annually or seasonally, what's your insights on like they respond to those like actively, like human become tolerant rather than being sensitive to those?
Yes, you have a couple hours. First, we have no experience yet that we can talk about publicly around bacteria, so I'm going to limit my comments to viruses, which is the only place where we have data to talk about publicly. I mean, the piece that we like a lot about mRNA is that if you think about it, most viruses, not all of course, are mRNA viruses. Which is why when scientists tried 40 years ago to make mRNA drugs before they tried to make recombinant, it didn't work because of things we know now, because we understand much better the immune system. Those animals we were testing 40, 50, 60 years ago in academic labs, they thought they got a viral infection. And so I think the piece that I think is really elegant is that you basically give instruction of a virus, so of a protein of a virus, but without giving a virus. And if you think about the CMV example, which again is a vaccine very close to our heart, actually I have a pictogram of it here, is when somebody gets naturally infected, the virus is going to use the cells that it's infecting to make protein to self-replicate using the ribosome of human. What are we doing? We're giving the instruction to a cell of a human to make, using the ribosome of a human, a protein that is a protein encoding the protein of a virus. So the piece that I think is very elegant about this technology for the application of infectious disease is this ability to mimic biology, because I'm a big believer in evolution and not trying to be too cute about biology. Biology is hard. And trying to really replicate what's happening with a virus, but without a virus. So you are in a very exquisite control of the quantity of proteins you get made, how it binds or not to the cell. So I think this is why this is going to be quite interesting approach for viruses. I cannot talk yet about bacteria. We're kind of working on it, obviously, but it's much more complicated than viruses. We good? Thank you very much.